So good afternoon, happy Friday. Yay, the weekend is here. So we're going to finish up this brain and evolution lecture. I think there's like four or five slides left over, and then we're going to start neuroanatomy. Yay. So don't be too intimidated. Yes, there's going to be lots of new terms that are sent your way. I often have students that say to me, how do I study? How do I remember all these things? Unfortunately, there's no magic mnemonic, particularly with these new terms. Really, all you're going to have to do is sit down and do some memorization. That said, it does get easier. Once you finish this course, you'll remember it for the rest of your life. Right, yeah, for sure. Okay, so what we ended up, the topic last class was the idea of relating brain size to intelligence. And I brought up the idea of how do we can look between species, but it's very difficult to make comparisons within species. So given that mice brain is a given weight or volume or what have you, does that mean that that is related to, in any way, to my intellectual capabilities? So how do we control for skull thickness, volume versus weight? And then we ended on this idea of body weight. Yes, men do generally have bigger brains compared to females because of their bigger body size and greater muscle to fat ratio. But keep in mind that an individual's body weight can fluctuate over time, and we can have very tall guys and very small guys, very big guys and smaller guys. So it's really hard to use strict factors of body weight to really make any gross assumptions about intelligence. In addition, we have to control for factors like age, physical health, possible brain injury. All these things make it very, very difficult to draw any comparisons between brain size and intelligence. In addition, we have a major problem of causality, right? You as psych and neuroscience students, you've learned many, many times correlations do not equal causations, right? And so, for example, if we do have a large brain size, which relates to greater intelligence, how do we know that the direction of the relationship is not in the opposite direction, right? How do we know that it's not having greater intelligence means that your brain ends up getting bigger? We cannot determine that. In addition, there are other variables that may mediate or moderate this relationship, right? Things like nutrition, stress, child rearing, like how your parents were they fantastic parents, horrible parents, the kind of school you went to, what university you went to, how many classes you took in brain intelligence. So all these other variables may very well intersect with the relationship between having a large brain size and greater intelligence. The problem is also related to how we measure intelligence. You guys have taken Psych 1001, Psych 1002, you know that we talk about the idea of intelligence and how difficult it is to put that to a specific number. Yes, we have IQs, but as you probably learned, IQ is not a perfect relationship. What if you're really intelligent when it comes to comedy or physical capabilities? We can't really define that. In addition, people vary enormously in their own individual abilities, and this may depend on the task, right? So we might have person A that has excellent math capabilities but poor spatial abilities, and then person B might be the opposite. They may have poor math abilities and excellent spatial abilities. We would both probably describe these people as being intelligent, but how would that exactly relate to their brain? And when do we say, oh, well, math supersedes spatial, or spatial supersedes math? Some people would argue for one versus the other, right? So there are a number of fallacies when we start to compare the brain size with intelligence. They do not seem to be particularly related. This is what has been coming out in the literature. For example, it's been shown that people that have who are very smart have average-sized brains, and an example of this is Albert Einstein. Yes, certainly it's been shown that, you know, layer 5 of the planum temporale, which is thought to relate to math capability, is slightly elongated in the left hemisphere of Einstein's brain. Again, problem of causality. What if the many, many, many years spent thinking about physics and math led to that change in his brain? So we can't really make these conclusions. In addition, as I mentioned last class, women's brains weigh about 10% less than men. But yes, it's true, our intelligence does not differ statistically. Okay? So women and men are equal intelligence, but our brain sizes are smaller. An interesting idea is that of the role of culture and how culture has really played a role in the evolution of our brain. So our large brains have allowed us to also develop a rich culture. And culture can be defined as complex learned behaviors that are passed on from one generation to the next through teaching and learning. A lot of what we do in our day-to-day life is culture. Being here in a university, that's culture. Playing hockey, culture. The arts, music, culture. You look at any other species on the planet, arguably some people would say that they have cultures, but they're not nearly as complex as human culture. In addition, our brain is highly flexible. We perform many tasks today that our brains were not originally selected for during early evolution. Think about our ability to program computers, play with our iPhones, do trapeze and Cirque du Soleil. Our brains, as they are today, arguably the greater part of the evolution occurred in the Paleolithic. So we're walking around with Paleolithic brains, yet we are still able to do all these complex tasks. That's not to say that there haven't been changes to our brain. Certainly there are. But the gross anatomy of the brain has not changed significantly in those millennia. So the question then, if we're not thinking about relating brain size to intelligence, is does having a bigger brain, is this actually better? The cost to this, and now I'm thinking more about EQ, right? Encephalization quotient. The costs is we have a long gestation period. For women, we hold our babies in utero for 40 weeks. That's an awfully long time when you think about rat gestation is 21 days. Yes, there are other species as well, like I think elephants, it's something like close to 2 years. But relatively speaking, we have a very long gestation period. Why? So we can have that time to have most of the delicate neuronal connections that are formed when the baby is most protected. Because as soon as the baby comes out into the world, it's much more susceptible to dangers. We are also not a very precocious species. A precocious species means like Bambi. Baby's born, boom, within an hour starts walking and can fend for itself. You leave a human baby that's just been born alone, it will die, guaranteed, right? So what this means is we have a prolonged dependence on our parents. Some would argue that even more so in this current era, right? It used to be that as soon as you reached adolescence, that's it, see ya, right? Now we're having the boomerang generation. We graduate from university, boom, back with mom and dad because we can't find a job. So we stay in their basement and boy, does that suck, right? No one wants to do that. Having a big brain is also metabolically costly. What that means is that we require a lot of nutrients and oxygen in order to keep our brain going. Our brain can only function, the main fuel source for the brain is glucose. The rest of our body can function on things like lipids. And this is why what happens when you're hungry, if you don't eat, the first symptom that you noticed is you're cranky, you're irritable, right? Because your brain is running low on glucose, low on fuel, and you get lightheaded, you get dizzy, you can't put thoughts straight together. So that's why you need to feed your brain. And because it's metabolically costly, we have to have the appropriate nutrients and oxygen in order to feed that brain. In addition, if you know anything about the human genome and genetics, you will know that we have many, many complex genes with complex polymorphisms, meaning there's more than one gene that contributes to a given behavior or function. And these are very susceptible to mutation. Right? Very rarely in other organisms do you see the number of genetic mutations that you do as with humans. You can probably think of a few off the top of your head, things like Down syndrome, right? You have an extra copy of gene 21. Kijcha syndrome, Huntington's disease, a lot of genetic mutations that babies never really survive past their first year of life, right? So complex genes, more susceptible to mutation. So a lot of costs to having these big, beautiful brains. That said, there is a good thing about having our nice complex brains that make us highly intelligent species. So one might argue that, yes, smarter is better. The benefits to this: increased survival and ability for group interaction. Last class we talked about how having these complex brains meant that we could form large communities and networks. And kinship theories would argue that then this means that you have a greater chance of survival because you have other kin that are looking out for you. Innovative behaviors such as tool use and social learning clearly propelled Homo sapiens, or whatever, Homo erectus, Homo habilis, into massive changes and ability for survival because of these capabilities. And this will predict success in a novel environment. So there's been experiments that they show that if you take birds or so some species of birds, other species of monkeys that have the ability for tool use, and you put them in a novel geographical environment, and you compare that to animals that don't have advanced tool use or cognitive capabilities, the species that have these advanced cognitive capabilities are more likely to survive. They can adapt to their environment. And this is why, where are humans on the planet? Virtually everywhere, right? We are not restricted to a specific part of the Sahara Desert or the grasslands in Africa. We are virtually everywhere, save for the poles, more or less, right? In addition, sexual selection pressures can do great things, right? We talked about the peacocks and their massive fanned tails with very pretty colored feathers. This allows us to be highly artistic and creative and more likely to attract other mates, and therefore more likely to pass on your offspring. An example of this is a species of bird known as the bowerbird. Do you guys know the bowerbird at all? Anyone? Yes? Yeah? Do you want to say a little bit about what the bowerbird does?
Oh, they're just amazing. I think they're native to Australia. They can be found only there.
Yeah.
And the male engages in this fascinating behavior of building a nest by picking generally, color relevant, like how shall I say, color appropriate objects, feathers, leaves, fruit, and such, just to attract the female bird.
Yeah. So they create this massive, it's called a bower, and it's literally a sexual arena. They are attracting women. They're saying, hey, or women, females saying, come here, come check out my sexy tent. So they've got like other feathers. They collect lots of things from the environment, and they make these beautiful sexual displays to attract the women. And the bigger and the better the display, the more likely that female is going to mate with them, and therefore the more likely their offspring get passed on, and that gene to produce those complex environments continues. No. Wren.
Yeah. I'm just wondering, would that be the same bird? I'm thinking of a bird that makes a nest like what you were saying, but everything blue.
Yes, that is the same. Yeah, that's the bowerbird. So often they'll, and you can see here, this is a picture from another textbook, that they've collected a lot of, sorry, I have to keep reminding, they've collected a lot of blue objects. Now, anyone want to take a guess as to why blue? Why blue? Yeah?
It's not common in natural environments.
Yeah?
Because birds are relatively colorblind, so they can only see certain colors of blue.
Yeah, so it's more that their ability to perceive color is limited to the 350 to 500 nanometer wavelength, which is related to the color blue. So obviously I'm going to, some species, other species they see red because they want to see the berries, right? So I'm going to show you a video of another, this is like one of my favorite videos ever, just to show you, like, and it has nothing to do really, like you're not going to be tested on this, it's just out of interest to show you the complexity of behaviors in other species. If anybody's seen this, I'm sure you'll quickly recognize what I'm showing you, but it's a video of David Attenborough. Anybody know David Attenborough? He's like the man and he studies all animals. And he's watching a bird, species of bird called the lyrebird. I'm just going to ensure that we have sound here, because sound is critical. So this species of bird, it is kind of like a mimic. What it does is it hears all the other bird calls in the environment, and it stores it into its musical repertoire or song repertoire. And essentially, this is what they use to attract other females. The greater the repertoire of sounds, the more likely the female is to mate with that bird. So you'll see in this particular bird, he's not only replicated other birds, but other sounds in the environment. So I'm just going to make sure this is...
What bird has the most elaborate, the most complex, the most beautiful song in the world? Well, I guess there are lots of contenders, but this bird must be one of them. The superb lyrebird of Southern Australia. He clears a space in the forest to serve as his concert platform. To persuade females to come close and admire his plumes, he sings the most complex song he can manage. And he does that by copying the songs of all the other birds he hears around him, such as the kookaburra. It's a very convincing impersonation. Even the original is fooled. He can imitate the calls of at least 20 different species. He also, in his attempt to outsing his rivals, incorporates other sounds that he hears in the forest. That was a camera shutter. And again. And now a camera with a motor drive. And that's a car alarm. And now the sounds of foresters and their chainsaws working nearby.
Pretty cool, huh? You ladies attracted? You interested? So, you know, it'd be nice to have a discussion on this, but you can think and try to relate this to humans, right? What kinds of things do human males do to try to attract females? How do they try to increase their repertoire? Dance moves, guitar moves, etc. All right, so we're going to switch gears altogether, and we're going to move now to our first real, what I would say, is the content-based lecture for this class, and it's on the gross neuroanatomy. So I have brought, every professor brings their brain with them. So I'm going to talk you through a little bit of the gross neuroanatomy of the nervous system. Not just the brain, because the brain is only one component, probably the major component of the nervous system, but it is only one component. So first of all, we're going to talk about how the brain is organized, or the nervous system is organized. And you can look at the organization of the nervous system from an anatomical and a functional perspective. So for you guys in 1201, these slides are familiar to you. So first of all, we have the nervous system. which can be subdivided into the central nervous system, which comprises the brain and the spinal cord. And then we have the peripheral nervous system, which is anatomically subdivided into both the somatic and the autonomic nervous system. The somatic nervous system are those nerves that control things like your muscles. This is by contrast to the autonomic nervous system, which has nerves that control things like your inner organs, your gut, your heart, your liver. It's called the autonomic nervous system because it is autonomous from your control. You cannot send a signal from your brain saying, tummy, please digest today's lunch, right? But you can send a signal from your motor cortex to say, extend my right arm. And that is done via your somatic nervous system. It's your peripheral nervous system because these are your peripheral nerves. They're the nerves that extend out of your spinal cord or out of the cranial nerves of your head, which we'll talk about shortly. So that's how we can look at the nervous system from an anatomical organization. We can also look at it from a functional organization. So functionally, again, nervous system at the top of the hierarchy, subdivided into the central nervous system, again, which comprises the brain and the spinal cord. And we say that the central nervous system is what's responsible for mediating behavior. Walking, talking, thinking. Now we have the somatic and the autonomic nervous system here that are further subdivided. So that somatic nervous system, which transmits sensations and produces movement, is subdivided into your cranial and your spinal nerves. Your cranial nerves we'll talk about in a few more slides. The autonomic nervous system is responsible for balancing internal functions. So as I mentioned, it's responsible for things like digesting your lunch, increasing your heart rate. And this portion of the nervous system is further subdivided into your sympathetic and your parasympathetic divisions. Your sympathetic division, you probably remember learning this in Psych 1001, is responsible for what we say is fight or flight. It arouses the nervous system to prepare the organism to deal with any potentially stressful encounters. So you see a bear running towards you in the forest. Your exam is in 2 hours and you haven't studied. What does your body do? Your brain sends a signal to these nerves that are involved in the sympathetic division, and it says, produce adrenaline and panic or run or fight, right? So it has a metabolic portion, but it's an adaptive response to help the organism deal with stressful encounters. The sympathetic division, it's like a seesaw with the parasympathetic division. The parasympathetic division is the rest and digest, right? So most of the time we're in that state, right? So when you're running away from the bears, you're not going to be wanting to devote energy to digesting your lunch. You want to devote the energy to your muscles so you can run or fight. So the rest and digest, the parasympathetic division, is that calming portion that when the organism is not in these stressful situations is focused on the sort of the normal everyday functions of the body. In later lectures, we'll talk about a little bit about stress and what happens when you are frequently putting your nervous system to an extreme where you're always in that sympathetic aroused state, how that can actually lead to disease. Things like coronary heart disease, heart attacks, immune disorders, etc. The way I like to think about this, to remember the difference between sympathetic and parasympathetic, I always say, think about the parasympathetic like parachute. It's trapping energy or air to slowly bring you back down to ground. So if you're trying to remember the difference between sympathetic, parasympathetic, think parachute, parasympathetic, capturing energy and bringing you down to the ground. So in the functional organization, we're focusing on how the parts of the nervous system work together to ultimately cause behavior. Also, two different terms that you should know is the difference between afferent and efferent nerves. So afferent nerves are nerves that are coming into the central nervous system. So things like the nerves that's connected to the fingertip, right? We're going to bring sensory information up via a number of different synapses up into our arm, into our spinal cord, up into our brain. Efferent is outgoing. So efferent, think E, exit. And these are nerves that are coming out of the central nervous system into the periphery. Okay? So sensory information coming into the CNS, afferent. Efferent is motor information leaving the CNS. And these spinal nerves here, they can be either afferent or efferent, right? We have afferent nerves that are controlling our muscles, but we also have afferent nerves that are controlling our muscles, but afferent nerves that are connected to our leg that say, oh, somebody's pinching your leg, for example. We need to transmit that same information. So don't be confused. Your spinal nerves can have both efferent and afferent components. So we need to know a little bit about, because we're going to be talking a lot about anatomy, and we need to be familiar with some of the terms that are used when we talk about different structures in the brain. So here, this is a really creepy photo of a guy whose head has been cut open. And they're basically delineating brain in 3D space, right? So when we are talking about structures that are very close to the front of our face, we would call these things anterior. So anterior towards the front. Anything that's towards the back of the head would be considered posterior. So anterior towards the front, posterior towards the back of the head. Okay? So for example, I might say that the cerebellum is located posterior to the frontal cortex, because the frontal cortex is at the very front part of my brain, and the cerebellum is in the very back part of my brain. So that's how you're going to hear these terms being used. The other direction is from side to side or towards the middle. So things that are towards my ears would be called more lateral, to the side. Things that are more towards the middle of my brain would be referred to as more medial. So middle, medial. Okay? So my corpus callosum is medial to Heschel's gyrus. Don't worry about what those things mean, but I'm just saying that's what, in papers, this is the kinds of things that you hear. The final direction is up and down. So at the top of my head or towards the bottom of my head. And towards the top of my head, we call that dorsal. Towards the bottom of my head, or the bottom of the brain, this is ventral. Okay? So top, dorsal, bottom, ventral, anterior, posterior, lateral, lateral, towards the middle. Look, I can, woo, crazy. This is medial. Okay? So obviously, some of you who are a little bit more astute will say, okay, well, dorsal means back, and ventral, vent, means stomach. Why are we referring to the top of my head as my back and the bottom as my stomach? Well, you have to think about what we are as humans compared to other species on the planet. We are bipedal. We walk around on 2 feet. We evolved from a common ancestor that would have been a quadruped, walking around on 4 feet. When you look at these in terms of directionality of the brain of a dog, for example, it kind of makes sense. The top of the dog's head is also in the same plane as its back. The bottom of the dog's head is also in the same plane as its stomach. Because the neur axis, and that's a fancy way of looking at the brain connected to the spinal cord, is all parallel to the surface of the ground. In humans, our neur axis is tilted at 90 degrees. Right? So our dorsal is no longer in the same plane as our back. Make sense? Anyone confused? You guys at home? Talk to me, Marigold. All right. This last slide also shows that you can also use terms like rostral and caudal. And this comes from like when you're thinking about birds, because rostral means the rostrum is the beak and caudal is the tail. So sometimes they can be used interchangeably with things like posterior and anterior, but for humans, we're going to stick to posterior and anterior. In addition, superior and inferior are used interchangeably with dorsal and ventral. Okay, so this is the kind of thing that you need to know in exam. I might give you a blank slide like this with anterior and posterior ghosted out, and you need to tell me what direction it is.
Do you want us to know the birds as well?
Is this in your textbook? Yeah. So if it's in your text, then yes. I believe this is. All right. In addition, you're going to be looking at lots of different views of the brain, not only just the outside and not only just describing different structures with relation to one another. So I'm sure some of you, you're probably tuned in to neuroscience even before you took this class. So you may have read studies in the news, and you may have seen pictures like what we're showing you in the screen here. And these are MRIs, and don't worry, we'll learn about MRIs in Chapter 5, but basically it's a scan of the brain that uses magnetic energy to differentiate structures of different sections of the brain. And you can see they look very different, right, on gross inspection. So this kind of view of the brain is known as a frontal view or a coronal section. And when we see this, it means that I have taken a slice of the brain as though I was cutting from the top of my nose to the back of my head. So you can see here, it's perpendicular to the surface of the ground. Okay? So I would have taken a slice of the brain here, and this is what I would have seen. Okay? This dorsal view is what would happen if I took a slice that was parallel to the surface of the ground. This is a horizontal section. No surprise, it's a horizontal line. And a medial view is what would happen if we took a sagittal section, so if we sliced between my ears, right? Like that. Anybody want to say or hazard a guess as to why would we want to represent the brain either frontal view, dorsal view, or medial view? What's the point? Why would you see an article that shows it from the frontal view compared to the medial view? Anna?
So that you can look at all different portions of the brain.
I'm gonna tie up your answer a little bit. So she's saying to look at from the brain at all different points. I'm going to say if we're interested in a particular structure of the brain that can only be seen from a medial perspective, we're obviously going to show that. So for example, in the last slide here, we very clearly see what's called the corpus callosum, which you guys can see here. I don't know if I should be like this or like this. But the corpus callosum is this white line here. This is what's called the corpus callosum. It's the main fiber tract that connects the two cerebral hemispheres. We can really clearly see it in this medial view. We don't really see it in either of the other two views. Right? So depending on what we want to represent or show in our figure, that's the view we would show. Okay? So in your textbook, this is a summary slide. I'm just cueing you to it. It's a table. It goes through every single thing I just discussed. Right? So if you need, for those of you that are visual, study using the diagrams. For those of you that are verbal or textual, I should say, you can study these definitions. Okay? That was the only point of showing that slide. So now moving right into the nitty-gritty of what's going on with the brain. The first thing that you should be aware of is that our brain is located in our skull. Right? The skull is that kind of thin, bony structure that encases our brain. We talked about it last class in that we could only grow our brain to a specific size because we were limited by the size of the skull. Our brain is not sitting directly underneath the skull. There are three very specific layers that protect the brain from the surface of the skull. And these are called the meninges. They are three layers of protective tissue. So you can see here, going from the surface of the skull towards the brain. in this picture here, you can see here's the skull. The first layer that we see is the dura, and we refer to it as the dura mater, which translates literally to hard mother. Why mother? Well, interestingly, little fun fact, the first individuals to describe the meninges were they were Arabic speakers. And when they were writing down the description of what they saw, they used I think. Anybody speak Arabic? Yeah? It means mom. Yeah, but it was a mistranslation. And actually what they were trying to write was membrane, and they ended up writing mum. And so it got mistranslated down the years, and now we actually use the Latin form, which is mater, which means mother. So do you know what membrane is? No. Okay, so we just know and we're good. Okay. So this is the tough, it's called hard because it's the tough outer layer. Anybody heard of a subdural hematoma? Right? You watch ER. I've got a subdural hematoma. Hematoma, fancy word for a bruise. Like if you bruise yourself, you can say, I've got a hematoma, and get out writing your exam. Just kidding. Don't do that. A subdural hematoma is when you actually have bleeding. Sub means beneath the layer of the dura mater. Right? So which is really bad, because what happens is when you got bleeding, you increase intracranial pressure, and that is just not good for your brain. So we've got the dura mater here. The next layer is the arachnoid layer. And arachnoid, anybody is arachnophobic, you know what that means. Arachnoid means spider. And it's called that because it looks like a web. So it's got all these fun little separations in it. Finally, the next layer is the pia mater, or soft mother. And this is the membrane that adheres to the surface of the brain. So it literally covers all the surfaces, the grooves and dips in your brain. And another thing that you should know is that beneath the arachnoid here and the pia is circulating cerebrospinal fluid. So beneath the arachnoid and the pia is the cerebrospinal fluid. And one of these spaces has a specific name. It's called the subarachnoid space. So literally beneath the arachnoid layer is the subarachnoid space, and it's filled with cerebrospinal fluid. So what is cerebrospinal fluid? Here is another picture that I have taken from the mighty Google, which is not in your notes. And you can see in blue here, this is all the circulating cerebrospinal fluid in your brain. And you can see it is in these subarachnoid and subpial spaces here. And it also is located in parts of the brain. And its main role, it consists of sodium chloride and other salts. It fills what are known as the ventricles, which I'll get into shortly, and circulates around the brain and the spinal cord, and it cushions the brain. So if you have head trauma, which is sadly can be quite common, this is one more mechanism aside from those meninges that can protect the brain and cushion it. Okay, so it's like putting your brain in a bubble wrap. Anybody heard of meningitis? Can be lethal, meaning you can die from it. This meningitis, anytime you see the word itis at the end of the term itis, it means an inflammation of. And meningitis is what happens when you have an infection of the meninges and the cerebrospinal fluid. You can have both bacterial or viral meningitis, which reminds me of a song by Cake, I think. Viral meningitis got me down. Anyone? No? Okay. Bacterial meningitis you can treat with antibiotics, although bacterial form is more deadly than the viral form. And why it's deadly is because you've got this infection surrounding your brain. You've got inflammation of these tissues, and again, this can increase intracranial pressure, and not only can that cause your brain to run out of space to breathe, for lack of a better word, but this can also mean that you can have septic infection because it can get into your bloodstream. So when we look at our fun brain, And don't you all wish you could take your brain out of your head and examine it that way, because that would be so much easier, right? So rather than that, we've got this fancy brain here. And so looking at this brain, what you will first notice is the fact that the greatest part of your brain is all of this here, the colored portion, and that is called the cerebrum. And it's the major structure of the forebrain here, and it consists of two virtually identical, and virtually, we will explain later, hemispheres. So you have two sides of your brain. You have your right side, and you have your left side. And remember last class, I mentioned that the right side of your brain controls the left side of your body, and the left side of your brain controls your right side of your body. Okay? This cerebrum is the most recently evolved structure in humans. You remember I said that our brain evolved from the back to the front, so we kept adding on more and more to the top part of your brain. So this is the most recently evolved. So the next thing that you'll see is the cerebellum, or little brain. And this is what's in gray in my brain here. And you can see it on the slide. I should probably just stay here. This is the cerebellum here. And so the cerebrum basically sits on top of the cerebellum. It's called the little brain because in lesser evolved or earlier species, they really only had cerebellum and then maybe a little bit of the forebrain. It's involved in a number of things, but we'll limit it to being involved in coordination, so fine motor control, and possibly other mental processes. It's now receiving more and more attention for possibly playing a role in disease, for example. And we'll learn a little bit about the cerebellum shortly. The final thing, which is hard to see on my brain unless I take it apart, is something known as the brainstem. So in your textbook, they talk about your brain being like a fist on your forearm, and the brainstem is akin to your forearm. I don't know, can we zoom in on this? So I think this will be easier. So this portion in green here, this is all the brainstem. So it's also, like I like to say, it's like, oh, don't look at self not there. It's like a lollipop on a stick, and the stick would be the brainstem. So all of this is the brainstem. It's involved because, again, the brain involved from the back to the front, with the brainstem first, then the cerebellum, and then the forebrain. This is involved in the most basic functions, right? So things like breathing and respiration and heart rate are all controlled by structures that can be found in the brainstem. So what we're going to do throughout the remaining class is we're going to take you through the forebrain, parts of the cerebellum, the midbrain, and the hindbrain, or the brainstem, and what are all the different structures that can be found in each of these divisions, right? So in this slide from your textbook, just to make it more patent, this portion here is your brainstem. All of this, and you can see that the forebrain is kind of sitting on top of your brainstem with your cerebellum attached there. So what we talked about last class was the fact that the surface of the brain is heavily convoluted. The bumps of the brain are called gyri, so the singular form is gyrus. And the dips in the brain, or grooves in the brain, are called sulci, single form sulcus. And I'm not going to force you to do this, but every single bump and groove has a name, right? So the post-central gyrus, the post-central sulcus, et cetera, et cetera. So you can imagine anatomists have taken this and labeled every single gyrus and sulcus in the brain. And that's because some of these different regions have very specific functions, right? So we know that the post-central gyrus is involved in motor control, for example. Okay, and I forgot to mention as well that the cerebellum is obviously heavily convoluted as well. So you see sulci and gyri in the cerebellum. A fissure is a fancy word for saying a very deep sulcus. So there's something known as the Sylvian fissure, which is located here, and this is in particular quite deep groove. So when I took brain and behavior in 3rd year, I had to memorize all these things, and no surprise, I did not do well in the class because, yeah, too much. Too much. So another thing that you should be aware of is that we've talked about the metabolic demands of the brain. Your entire brain is covered. It's heavily vascularized, meaning it's covered with arteries. And these arteries are what feeds and nourishes the brain, right? Because we carry important things in our bloodstream, like the glucose, right? Gets into the bloodstream and is carried via the bloodstream into the surface of the brain. And we have very special cells that are responsible for transporting the oxygen and the glucose and all the other immune markers from the bloodstream into the brain. So just the major arteries are, for example, the anterior cerebral artery. Remember, anterior means towards the front. So this is a dorsal view of the brain, like I'm looking at it from above. And you can see now here from the lateral view what the anterior cerebral artery basically feeds and nourishes what's ghosted here all in red. Right? So these neurons are fed by the anterior cerebral artery. The middle cerebral artery, here's the lateral view, feeds these neurons and a little bit of the bottom of the brain here. And the posterior cerebral artery is here it is. It feeds mostly the inferior surface of the brain and some of the inside medial view of the brain. What this means is neurologists can very quickly determine, without having a scan of your brain, where a stroke has occurred. And a stroke is essentially a broad term to say that you've had damage or a block in your bloodstream, and it's preventing that particular branch of the middle cerebral artery or what have you to nourish those neurons. And if you're not getting the oxygen and you're not getting the glucose, those neurons will die, right? So if you have a clot, you can, smoking can do this, lack of exercise, and the clot forms in that specific branch of your artery, those neurons will die off. And because we know that brain tissue relates to brain function, if suddenly you have the lack of ability to form speech, we'll know that, for example, it's probably some branch of the middle cerebral artery because that feeds the parts of your brain that control language. Okay? Any questions? All right, so a little bit about the internal features. We've looked at the outside of the brain. Let's now delve in. You've probably heard the terms gray matter and white matter, and it is descriptive in terms of its color because we do have gray and white matter in our brain. Here is what kind of slice. What kind of view is this? Either one I'll accept. We've taken it from this plane. What view is it? Coronal. Coronal, that's right. I also accept frontal. So here in this coronal section, you can see that these areas of the brain are gray, and the inside here is white. Now the gray matter is gray because it consists of cell bodies. So cell bodies, or neuronal bodies, are gray. And the white matter is the axons. So what we'll learn probably next class is that the axon of the neuron, you would have all heard this in Psych 1001, I'm sure, is covered with a fatty sheath called the myelin, and the myelin is literally white. So all of this white matter is just showing that all the connections between the neurons. Okay? A ventricle, which I alluded to earlier when we were talking about CSF, is a cavity in the brain that contains cerebral spinal fluid. We have 4 ventricles. Here, we have 2 lateral ventricles, so the left side and the right side. And you can see them here in this coronal section. You can also see here, this is the corpus callosum from this view. So a little bit more about the ventricles. Like I said, we have 4 of them, a left and a right lateral ventricle. Remember, lateral means side. We also have the 3rd ventricle, which to me always looks for some reason like a crow or a chicken or something. I don't know. It's got a beak here, an eye, and a head. I don't know. That's me. And here is the 4th ventricle, and they're connected via the cerebral aqueduct here. So here is a side view of the brain, and here's a view from the back. And you can see this is the 2 lateral ventricles, the cerebral aqueduct, which connects the 3rd and the 4th ventricle. As I mentioned, the corpus callosum is what connects, it's the main fiber system that connects the left and the right hemisphere. Here is the plane of cut. It's a sagittal cut, and you can see this is the corpus callosum, as I showed you in the previous slide. We can get some really wacky things going on when we sever the corpus callosum. And occasionally we can do that. It would be done in extreme cases, but if somebody has intractable epilepsy, which means they suffer from grand mal seizures that are not controllable via medications. And sometimes it's been shown that if you sever this tract for whatever reason, this can alleviate the epilepsy, but can lead to some really crazy things. And I don't have time to show you the video in this class, but I show it in my sensory processing class of a guy who has had a cholecystectomy, as it's called. And what happens is essentially you're creating 2 brains from one. And there's an example of a guy who had this corpus callosectomy. His left hand reached out to get a brown shirt to wear in the morning, and his right hand smacked it away and picked the blue shirt. Right? So you've got 2, I mean, this has whole implications for philosophy of mind and God and blah, blah, blah. But essentially this means that there's 2 separate volitional controls in the brain, right? It also has implications for vision, because the visual system is also crossed. So the my left visual field is processed by the right side of my brain, and vice versa. If you sever that, what happens is that you can show them a picture in their left visual field, and because that's processed in the right side of the brain, which can't get to the left side of the brain, which controls language, they say they can't see anything. However, if you ask them to draw it with their right hand, they will. So they say, I don't see a dog, or I don't see anything, and then they'll draw a picture of a dog. And then they'll be like, oh, that's a dog. Wacky. Isn't the brain cool? Okay, so moving right along at a very quick pace. We've probably heard this before. This is hopefully review for you, that there are 2 main kinds of brain cells. The most probably one that we've heard about is the neuron, and it controls most of our functions. These are the ones that communicate, and again, we'll learn a lot more about this, if not next class, then the class after. There are approximately 80 billion neurons in the brain, which is a whole heck of a lot, and I can't even visualize how many that is. An example of a neuron here, this is a very fancy scanning electron micrograph showing neurons in the cerebellum. And here is the cell body and the axon and the dendritic tree. And this is a cartoon version of what this neuron would look like. These are called pyramidal cells because their cell body is in the shape of a triangle or a pyramid, because we're in 3D. And they are mostly found in the cerebellum. So you can see here's the dendritic arborization and the axon here. Glial cells are sort of the younger brother, or the heir and the spare. They have a more supporting role in the nervous system. And whereas the neurons mainly communicate and mediate behavior, the glial cells are more assisting in the activities of the neurons. So they do things like nourish the neurons. They're the ones that make connections with the bloodstream, for example, and transport the important nutrients from the bloodstream to the neurons. They also insulate the neurons. They're the ones that form the myelin sheath, for example. They're also important in immune function. So if there's disease, they're the ones that help with the immune signaling. And here's an example of a specific kind of glial cell. Don't worry, you'll learn all about the different kinds in next class. This is an astrocyte. It's called an astrocyte because it looks like a star. So you can see this is the scanning electron. They really do look like pretty little stars. And you can see there's extensive arborization here relative to the cell body. Yeah?
Are glial cells found anywhere other than
Yeah, they're found in the peripheral nervous system. Yep, they're found all over. Yep, widespread. So she asked, are they found outside the brain? Yes. And depending on whether it's in the brain, if in the CNS or the PNS, they have a different name for them. So a little bit more on terminology, just so we are all on the same page. When we talk about anatomical regions in the brain, sometimes we will say things like this particular nucleus. And a nucleus is a fancy way of saying, and this is the plural is nuclei. It's a group of cells that are clustered together that anatomically are distinct. So if we stained, like took a slice of the brain, exposed it to a dye, we would see that there is a very specific anatomical delineation of a population of cells. We refer to this as a nucleus. So it's clusters of cells that anatomically are unique, and oftentimes, more often than not, they form a very specific function. So I always give the example, because it's always in the top of my head, of the nucleus accumbens. For those of you that took addiction with me, you remember that the nucleus accumbens is a very important structure that's found in the brain that mediates the process of reward. So whenever we feel really good, we eat a nice big juicy Big Mac, we have sex, we drink tasty drinks, we play music, nucleus accumbens releases a very specific neurochemical. A nerve is a grouping of axons. So we've all heard about nerves. Now today you finally know what exactly is a nerve, and it's a group of axons. So things like the optic nerve are all the axons that are collected from your retinal ganglion cells in your eye that bundle together and form the optic nerve, which exits the back of your eye and makes connections with your thalamus. We usually use the word nerve to refer to these axons that are outside of the central nervous system. A tract is a very large collection of axons that course together in the central nervous system. So what is an example of a tract in the central nervous system? What's connecting these two? Corpus callosum is an example of a tract, right? Because it's these axons that connect the two cerebral hemispheres. Because we've been talking about evolution and how evolution has played a significant role in the development of the nervous system, I just want to again highlight the fact that the human brain is quite complex. Yet it still retains some of the features of less complex brains, right? We didn't just reinvent the brain with every new evolved species. That would be very, very metabolically taxing, right? So rather than bring an entirely new structure into the brain that controls breathing, for example, we retained the old structure, but the new structures, now we have new connections to those regions, if that makes sense. So we still have our lizard brain. I always say sometimes we make decisions with our lizard brain, right? Because the lizard brain is the very back part of our brain, and it is still there, right? We just have our lovely, nice frontal lobe, which tends to inhibit some of our lizard brain impulses. And this keeps us, this is what I say, what separates us from the rest of the animals, right? So we don't necessarily see a hot guy in the street and yell out, "Hey, baby," right? No, no. Our frontal lobe, thankfully, prevents us from doing such silly things unless we're under the influence of our good friend alcohol. And then alcohol gets into your frontal cortical neurons, shuts her down, and says, yes, it's a good idea to show my boobs to a video of Girls Gone Wild, right? To get some beads or whatever. That was a huge diversion. Wow. Okay, so all that is to say that because of this, because we have all these complex functions that are added on to the back part of our brain, most behaviors are not the product of a single locus in the brain, right? yes, there are specific brain regions that are involved in specific things, but you should know that most behaviors are pathways or loops in the brain. One region of the brain is activated, which then goes to another region of the brain, which then makes connections with yet another region of the brain that probably goes back to the original brain region and probably has an inhibitory connection. So there are loops of neurons that are involved in complex behaviors. And that's why it's highly conservatory, right? It wouldn't make sense if just this one little piece of tissue controlled breathing, or this one little piece of tissue controlled sight, right? We need to have some conservation built into the system. So here's a slide that if you ever take neurodevelopmental determinants of health, which is a third-year class, you'll get to know this figure very, very well, because it's an embryological description of how the brain develops, both in an individual and as a species. Has anybody heard the phrase ontogeny recapitulates phylogeny? Ontogeny recapitulates phylogeny. Anybody knows Latin, they can probably figure this out. But Latin is a dead discipline, so probably not. The development of the individual mimics the development of the species. As our brain develops within us, the brain development mimics how it developed in the species. Right? So if you look at lower organisms down the evolutionary pole, you'll see that their brains look like what our brains look like when we're very young. Ontogeny recapitulates phylogeny. Development of the individual mimics the development of the species. This is why in utero, what do we have? What do we have in utero? What do we look like? Birds, lizards, right? Anybody seen like a fetus, an embryo? Little tails, right? So the development of the individual mimics the development of the species. So here, this is an example of a vertebrate, so not just human, but vertebrate nervous system, very early on, right? It looks, and I'm not going to teach you this until we get to neurodevelopment, but just know that we start out with, as a tube. That's what the nervous system essentially is, a tube of cells. And we have these three swellings. So at the back, we have tissue that eventually becomes the spinal cord. Towards the back end of the brain is something known as the rhombencephalon. Cephalon means head. This is known as the hindbrain. It eventually becomes the hindbrain. The mes, or middle encephalon, is the midbrain. This is that second middle swelling. And the prosencephalon, or first brain, is called the forebrain. So basically what's happening in this figure, and this is from your textbook, and it's showing you by color. The prosencephalon, very early on, becomes the forebrain, which then is further divided as we develop as a species into the telencephalon and the diencephalon. So here in blue, the telencephalon and then the diencephalon, which then, as a fully formed adult brain, comprises of all these different structures. Okay, so the prosencephalon, or forebrain, becomes further divided into telencephalon and diencephalon, and further goes on to consist of all these structures. The blue is what ends up being the forebrain. The mesencephalon, or the midbrain, here comprises these structures, which are part of the brainstem. The rhombencephalon, or hindbrain, comprises of the metencephalon and myelencephalon, which forms the cerebellum and other structures, again found in the brainstem. And the spinal cord is the spinal cord. Yes, a lot of new terms here, right? So developmentally, this is all you need to know is prosencephalon turns into these things, mesencephalon into these, etc. This is, I believe I remember learning this, it's alphabetical to some extent. So telencephalon, diencephalon, and then mes, met, my, they're all in order, right? MES, MET, MY. That's how I remembered learning them as an undergrad. Okay, so what we're doing today, this is all you need to know for now before we get to neurodevelopment, then you're going to need to know quite a bit more. But all I'm trying to show you is that these adult structures all formed from these early tissues, And what we're going to do for the remaining portion of the lecture is talk about how all the different divisions of the forebrain and the brainstem. So we're going to talk about all these different structures and what they do. So you're going to need to know their names and their functions. Again, I know there's a lot here that's new. You're just putting all of this into your mental vocabularies, but trust me, it will get easier. So we'll start with understanding the major divisions of the central nervous system, which, as I mentioned, comprises the spinal cord and the brain. And we're going to subdivide the brain into the brain stem and the forebrain. Okay, because remember, the forebrain is the major portion of our brain. The brainstem comprises of the midbrain and the hindbrain. So let's start out talking about the spinal cord. You probably have some fleeting understanding of what the spinal cord does. You're probably tuned into it because of things in the media where we hear about people who've had damage to the spinal cord. Christopher Reeve, Superman, great example. Fell off his horse and ended up with a break, a very high cervical break in his spinal cord, which meant that he was effectively a quadriplegic. He also had to be on a respirator because some of the neurons that are in the cervical division of your spinal cord control respiration and breathing. So he couldn't do that on his own anymore. So I believe, I don't know exactly what his break was. If somebody does know, please let me know. But essentially, your spinal cord consists of 31 pairs of nerves that extend out of your vertebrae. So this is a picture that is not from your textbook, which is why I couldn't distribute it. But here is your spinal cord here. It is the interface basically between sensation and movement, right? We have sensory nerves that come into the spinal cord, send it to the brain that says, hey, hot cup, or, ooh, that's a nice massage that I'm feeling. And then we have nerves coming out of the nervous system, the efferent nerves, that come down the spinal cord and make contact with the dermatome, the patch of skin that had that sensory nerve attached to. So we can control the movement. So it makes sense. I touch a hot cup, the efferent nerve coming out that's controlled by your motor system is going to want to remove that left hand, not your right hand, right? That makes sense. So it can act to some extent independently from the brain. And these are what are called spinal reflexes. So even if you have a break in your spinal cord, you can still maintain some reflexes. And an example of that is your knee jerk or patellar tendon reflex. And that's what happens if you've seen on TV. The doctor will come in with a mallet, a rubber mallet, and hit you just at the right spot. I could never do it myself, but they know. And you have a knee jerk reflex. So even if you have damage to your spinal cord, you still maintain this reflex. Because the spinal cord, and I should have made this even more patently clear if it wasn't already, all of the nerves that come out of the different levels of the spinal cord. So here we have the cervical division. It's called the cervical because it feeds the neck and the top of the shoulders. These nerves feed that specific part of your body, right? So the cervical division is the very top part of your body. The thoracic here, so we have T1 to T12. Those levels, or vertebrae, and the nerves are controlling your thorax, or your, I guess it's from your upper chest to your lower back. The lumbar division here, the L1 to L5, controls, I believe, from like your belly button almost down to your feet. And the sacral division here controls kind of like the backs of your legs and your butt. So depending on, like, if you've got damage here, you've severed these nerves, they can't regrow. This is why we need money and research to find out how we can regenerate spinal cord neurons. And Patrice Smith, who is a scientist faculty member here in our department, does that. Because these nerves can't regrow, it means that you have no input or output from that part of your body, right? So if I have damage up here in my neck portion, that means that everything below that, we can't feel, right? That's why you would be effectively a quadriplegic, because you're not getting any input from anything below your neck. A paraplegic would be, depending on the break again, somewhere a break in the thoracic region, right? Because a paraplegic, you can still move your arms, but you can't move your legs. Is that clear? I always have a hard time describing that because I don't really know where to begin. So that's your spinal cord. Let's start at the back and talk about the brainstem. So here's that figure from your textbook that I said that makes the analogy that the brainstem is like your fist and your forearm, where the fist portion is analogous to your diencephalon, the wrist can be analogous to your midbrain, and the forearm is analogous to your hindbrain. So here you can see it's like I popped off, and I think I can do this. Da, 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 da. I popped off the cerebrum, and I'm left with this. So that is your brainstem, which includes your cerebellum, but it popped off. So here you can see it comprises the diencephalon, the midbrain here, which connects the diencephalon to the hindbrain. So this whole thing is your brainstem. It begins where the spinal cord enters the skull, here. So you can see that this would essentially be your cervical vertebrae, would just be making connections here. It produces movement and is responsible for creating our sensory world. Lower organisms have the brainstem. They're still able to make a sensory world. Remember Pierre Flourens and his chickens? He popped off pretty much all of the forebrain, and the chickens were still able to kind of move around and eat. So they're still able to have some capabilities for movement. The three major divisions: hindbrain, midbrain, and diencephalon. So we're going to talk about what's in the hindbrain, what's in the midbrain, and what's in the diencephalon. Sometimes it's helpful, I know students have done this in the past, and I have meant to, but always forget, to create like a hierarchical chart, right? So what's in the brainstem? These three things. What's in this? These three things, etc. So if you are a visual individual, you like to map it out that way, it probably could be helpful. So the hindbrain is, as I mentioned, evolutionarily the oldest part of the brain. It comprises of the cerebellum here, a group of cells. Remember, it's also the reticular nuclei. It's called the reticular formation. And it's kind of like this tunnel of cells that courses all throughout the length of the hindbrain here in red. So that's the reticular formation. We also have the pons here. This is the pons and the medulla. And the medulla is the most inferior portion of the hindbrain. So it's really the most evolutionarily oldest part. And it's no surprise because the medulla is involved in breathing. So overall, the hindbrain taken together, major role in control of movement. And we'll talk through each of these in turn. So the cerebellum, as I mentioned previously, is more in fine motor coordination, complex movements, and some cognitive functions. So here is a sagittal slice of the cerebellum. And you can see, again, it is called the little brain because it has all these convolutions. It also consists of white and gray matter and some subcortical nuclei. So when you're playing piano or you're cheerleading, doing some crazy stunts, what's helping you do that is the cerebellum. And as I mentioned, it can have some cognitive capabilities. We're just starting to recognize its role in disease as well. One of the theories of autism implicates the cerebellum. What's interesting is that the size of the cerebellum increases with the physical speed and dexterity of a species. So our good friend the sloth, they're super cute, but not exactly the quickest species on the planet, have a very small cerebellum relative to the leopard, for example, or even the hawk. So the size of the cerebellum increases with physical speed and dexterity. So that's kind of, through a comparative analysis, a way of certainly arguably saying that it has a role in movement. So cerebellum, part of the hindbrain, part of the brainstem. Brainstem, hindbrain, cerebellum. Also, again, we've got that reticular formation here in red. And its main role is in arousal. So it plays, when we get to the chapter on sleep, you'll see that there's a lot of neurons connections and pathways that start out in the reticular forebrain and extend all throughout, what did I just say? Reticular formation, and extend all out to areas of our forebrain. So heavily innervated with serotonergic neurons, serotonin, because we know serotonin is implicated in sleep. So it regulates sleep-wake cycles. The pons, which literally means bridge. If you're interested, and I've had students ask me this in the past, some people like knowing the actual meaning of the word, and it helps them know what that structure is, if that makes sense. If you Google neuroscience terminology, you will find a website that takes you through every single term and what it means. So like pons, bridge, amygdala, almond, et cetera, et cetera. So if it helps you, by all means. So it's called the bridge because it connects the cerebellum to the rest of the brain. And it's also important in movement. We'll find out it's also implicated in sleep as well. And the medulla, the very inferior or rostral tip of the spinal cord here. And it performs very vital functions, breathing and heart rate. So if you have stroke to the medulla, you're not in a good place, right? Yeah, probably not a good plan to have that happen if you can control it. Moving right along to the midbrain, and we'll finish here today after this slide. So here is what kind of slice? Da, 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 da. Huh? No, sagittal is like this. We're doing parallel to the surface of the slide of the horizontal. Excellent. There we go. We're all awake. Okay. This is a horizontal slice because the midbrain is really, you can't really point to it on the brain, right? It's a series of nuclei that kind of connect the hindbrain to the diencephalon. So it consists of a few important structures. The tectum, which literally means roof. So the tectum is the roof of the midbrain. It's involved in sensory processing and both visual and auditory because it contains things like the superior and inferior colliculus. The superior colliculus is important in visual information, and inferior is important in auditory information. So one of the first places where, like, you hear sounds that goes into your ear, blah, 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 cochlea, you'll learn all that later. And then we have the auditory nerve that ascends into the midbrain. And one of the first groups of nuclei that it goes to is the inferior colliculus. Yep?
Will we be starting to know?
Yes. Anything that's on this slide? Yep. Yep. I know. It sucks. We'll all be there together, sucking hard. Yep. So it's because it's so back in the brain, like these parts of the brain are not going to be involved in like, hey, this is an orange. These are things like, there's something moving in my visual field, right? So very, very basic responses. And orienting, right? The tegmentum here, so we've got the tectum and the tegmentum. And this is the floor of the midbrain. It's involved in movements of your eyes and your limbs, some species-specific behaviors like sniffing, for example, and some implication in the perception of pain. So remember, pain is important, right? Evolutionarily, it serves an adaptive role. We need to know if something's hurting us so we can avoid it. And so it makes sense that we have some pain neurons in the back portion of our brain. Have an amazing weekend. Study hard. Don't drink too much.